Hello, my name is Camila Castillo, and if you're watching this presentation, thank you very much for doing so. And thank you to the Cancéropôle Lyon-Auvergne-Rhône-Alpes for the opportunity to share my work. But before we get into the hardcore science, let me ask you a simple question. When you get to a crossroad and your main path is blocked, what do? Well, melanoma cells may give us some insight through the research I'm presenting today under an e-poster format, titled Switching to a Different Path, the IGF-1 Route in Melanoma Resistance. This project is conducted at the Institute for Advanced Biosciences in Grenoble as part of my PhD studies. Melanoma is the most aggressive form of skin cancer, and even if it only represents 10% of all skin cancers, it accounts for over 80% of all skin cancer deaths. Two main therapeutic strategies are used to treat patients with melanoma: immunotherapies and targeted therapies. The latter has represented a major breakthrough in melanoma treatment with the development of specific inhibitors of the mutated protein BRAF V600E, such as vemurafenib. By blocking this protein, vemurafenib is able to limit tumor cell proliferation. However, most patients quickly develop resistance to the drug, and for over 40% of patients, the mechanism of resistance has not yet been identified. Interestingly, it has been shown that interactions between the tumor microenvironment and tumor cells promote tumor progression and therapy resistance. Moreover, IGF-1 is part of the autocrine signaling mechanisms of melanoma cells that stimulate their proliferation, and its receptor, IGF-1R, is overexpressed in many cancers. Therefore, the goal of this project is to elucidate the mechanisms of resistance to targeted therapies that appear as a function of the tumor microenvironment in melanoma cells. And in particular, we study the role of the crosstalks between extracellular matrix receptors such as integrins and tyrosine kinase receptors like IGF-1R in resistance to BRAF inhibitors. In order to do that, we developed human melanoma cell lines resistant to vemurafenib through chronic exposure to the drug. We used the SKML28 and A375 cell lines that harbor the BRAF V600E mutations. After 3 months, we obtained cells that were able to grow in the presence of vemurafenib and cultured them in 2D and 3D to avoid artificial activation of integrins. We evaluated the sensitivity to vemurafenib and found that both in 2D and 3D, A375 and SKML28 resistant cells significantly increased their IC50 values compared to the sensitive cells, which validates the generation of vemurafenib-resistant cell lines. Then, we evaluated the expression and activation of IGF-1R in our cell models through Western blot, and we found that IGF-1R is overexpressed and overactivated in both A375 and SKML28 resistant cells. Then we had to identify what signaling pathways would be activated downstream of IGF-1R. Therefore, we treated the cells with the IGF-1R inhibitor lincitinib, and we observed that AKT is activated in resistant cells cultured in 2D and 3D, and that this activation is partially blocked in the presence of the IGF-1R inhibitor, suggesting that other signaling pathways may be involved in AKT activation. Interestingly, the co-treatment with vemurafenib and lincitinib significantly reduces AKT activation, supporting the relevance of combination strategies. Our results also highlight the relevance of 3D cell culture, in which we observe a reactivation of the ERK pathway in resistant cells in response to vemurafenib that is barely seen in 2D culture. Finally, treatment with lincitinib alone or in combination with vemurafenib induces caspase-3 cleavage in resistant cells. In conclusion, our results show that vemurafenib-resistant cells take a different road and activate the IGF-1R AKT pathway to circumvent BRAF inhibition. We found that AKT activation is partially blocked in the presence of an IGF-1R inhibitor, suggesting the involvement of other activation pathways like integrins that can interact with IGF-1R and modulate cell signaling. Finally, the co-treatment of resistant cells with vemurafenib and an IGF-1R inhibitor sensitizes melanoma cells to vemurafenib. I would like to thank our team at the IAB and the organizations that finance this project, and of course, thank you to all of you for taking this alternative road. You can find more information in the poster, and don't hesitate to contact me for any follow-up questions.